So, with DNA replication, it's what we call semi-conservative replication. Okay, and so with semi-conservative, basically what this means is that we're going to use one of the two strands to make new strands. Okay, so you use one of two DNA strands as a template. For new DNA. Kind of a crappy D. Okay.
What is it like to divide them?
Yep. And so that actually kind of leads into what I want to talk about. There are going to be two really important ones, but three total enzymes. Okay, and enzymes are proteins that basically do something inside your body. Okay. The first one we're going to have, and again, remember, pretty much anything that ends in ACE is an enzyme. Okay? And so the first one we're going to have is DNA helicase. Okay? And what DNA helicase does is it attaches to DNA and basically unzips it. Okay? So you understand how you have like your A and your T, or your C and your G that are together. They're combined in the middle by those bonds, either 2 or 3, depending on which one it is. Right? We're good with that. DNA helicase comes and it opens it up. Okay? So DNA helicase unwinds and unzips DNA so that both strands are open. Okay? And so that's all that does. Okay? Is it basically opens up DNA and makes it so that we can have two new strands get formed. All right? And so you're going to have your, which one's leading, which one's lagging? Yeah, so you're making it's made to the 3. Okay? So our 3 to 5 strand, okay? This is going to be opened up, so essentially it just kind of looks like this right now, okay? It's still attached back here. It does its whole goopy weird thing. Alright, but your 3 to your 5 strand is the easy strand, okay? This is called your leading strand. Alright? This one down here is a little bit more complicated. It goes 5 to 3. This is called the lagging strand. Okay? And so this is where we will take the second and probably definitely the most important of the 3 enzymes. Okay? So the second one is DNA polymerase. Okay? So what DNA polymerase does is it attaches to old template strand, reads it, and makes a corresponding new strand. And here's a really important part. It always goes always moves from 5 to 3 on new strand. Okay, so it's always making 5 to 3. Okay, so we'll go ahead, use 3 colors here. So right here, we'll have this. This will be DNA polymerase. Okay, and so let's just say we have on our leading strand, let's say this will go, we've got an A, we've got a T, we've got a C, we've got a A, we've got a C. Okay? And so what the DNA polymerase will do is it will move this way. Okay? So again, this is a 3, this is a 5, right? On the leading strand, it's 3 to 5. Well, it makes the mirrored strand on the opposite side. So what will this go from? If this one goes from 3 to 5, this one will go from 5 to 3. Okay, so reading it in the way that it always does. So 5 to 3. And so what will this new strand look like? What's going to go here? Chargaff's rule. T, right? T's go with A's. Here, I'll have an A. Here, okay, T, G. Okay, and this one, the leading strand, it just goes and it reads this really easy. Okay, it just kind of keeps chugging along, making new DNA. All right? Any questions about that one? Now, this one does a little bit different stuff because the new strand is getting made this way, right? Well, but it's opened up in a weird way that is going to make it more difficult. Okay? So on the lagging strand, okay, we are going to have little pieces of RNA that attach, and they're called RNA primers. So RNA primers attach at points on lagging strand. Okay, and so basically you'll have, well, I thought I had another color. All right, so we're going to attach, and I'm going to do it down here, okay? We'll have little strands. No, I should make it even smaller than that because RNA primers are only like 3 to 4 nucleotides, okay? They're very, very short, okay? And so we need to go this way with our DNA polymerase. So the DNA polymerase on lagging strand, DNA polymerase attaches at primers. Okay? And so it's going to attach right here and right here. Okay? And basically what this will do is it will makes fragments from primer to primer. Okay? And so it'll read and it'll make the new strand that'll connect those. Okay? And I'll throw another one over here. Alright? and it'll connect those, okay? And so these are moving from 5 to 3, 5 to 3, okay? And they're going this way, all right? And it's still making the mirrored image, okay? So again, let's just say C, A, G, T, okay? On our new strand, it'd be G, T, C, A, okay? And same thing up here, okay? So I'll just draw some boxes. Okay, and so we've got that. But this isn't a full strand yet. We still have those little pieces of RNA, and RNA is unstable, okay? So, RNA is unstable and must be, what's the word I'm looking for? Switched out. Now with DNA. Okay? And so we've talked about, I said that there were 3 different enzymes, right? We have our helicase, we have our polymerase, and now we have to use RNA ligase is what it's called, okay? And so basically all ligase does is it removes, go ahead Evan. Okay, it removes RNA primer and makes DNA there, combining fragments. Okay? And there is one, it's not super important, but we can put this in your thing. These different fragments, so that fragment and this fragment that do get connected, all right? We get rid of these and we connect them. But those fragments are called, because they were discovered by a guy with the last name Okazaki.
Oh, that's funny. Sounds Japanese.
It's because he was. They're called Okazaki fragments, okay? So it combines, so it goes and it's combining all Okazaki fragments, okay? And then when you're done with this, you have 2 new full strands of DNA, okay?
What do you know? Good deal.
And it's, again, this seems kind of difficult, but really it just unzips. This attaches and starts copying the other side. And then the lagging strand's a little bit more complicated than the leading strand. A couple more things happen, but at the end, DNA polymerase is just copying the other side and making 2 copies. So at the end, you have 2 copies, 2 brand new full strands. Each of them has 1 old strand.
Does it eventually make off and make like more strands on each one of those, or?
Yep, and eventually, I mean, your DNA is always replicating. You make like a new copy of your DNA. Well, your cells are constantly dividing. I mean, your cells in your body, you probably make a full new set of cells like every 12 to 24 hours. And so within that, you have to, when you're making that new cell during S phase, right, synthesis phase of interphase, you need to be making a complete new copy of your DNA, right? So really, you make a whole copy of every piece of DNA in every cell in your body once to twice a day. Now, cells also die. We're pretty much, you guys are making more cells than are dying. I make about the same amount of cells that are dying. That's why you guys are still growing and I'm not. Okay? And then once you hit, what is it, like 80? 80, it becomes really hard to build muscle in your body. You can build muscle up until about 80, and then at that point, it's, if you work out, you can kind of just maintain a certain muscle tone, and then at 90, continuously atrophy. But anyway, that's because you start having more cells dying than are being created. Yeah.
So like 50 years ago, why is like it started decaying earlier than now?
I didn't hear what? Why did people start to decay?
Like no, like 50 years ago, why would we die away sooner than we do now?
Medicine. Medicine's a lot better. We have
But like, were our cells the same?
kind of, yeah, but the way we took care of them was different.